Professor Luke O'Neill is going to talk to us about an overview of COVID-19 detection and possible interventions. And he needs no introduction. We all know Professor O'Neill, but also, of course, he's an excellent scientist and he's going to give us a bit of science and evidence now. I won't read out his lengthy CV, which has been provided to me here, actually, but I will say, as well as being an excellent scientist and communicator, he's actually a very affable guy, and we can speak to this guy and understand what he's saying. So, Luke, I'm handing over to you now. Thank you.
Great. Yeah, thanks. I'm going to share the screen. Is that okay? Just to get the slides up.
Absolutely. Thank you.
Real. Let me look now.
I will mute here now. I think, is that correct? Okay.
Great stuff. Okay. Now then. Great. Okay. Thanks very much. Thanks for the invitation as well to Anne for the come and speak today virtually. We're living in a funny world, aren't we? Shame we can't meet up more. It sounds like there was an audience there. Heard clapping. There must be an audience of some kind, I suppose. But I'm going to give an overview of COVID, and I'm going to talk about some of our own research. And we've begun working on COVID, of course, probably back in March, I guess. I've always had an interest in the inflammatory process for many decades now, in fact, and inflammation during sepsis was a big part of what we did. And of course, when COVID came along, then it wasn't that difficult for us to apply some of our knowledge around anti-inflammatory effects during an infection, especially during septic shock, into the COVID world, I suppose. So I'm going to give you some very recent data. Obviously, it's a work in progress. We've only just begun this, really. But I thought I might give you a quick overview on the COVID-19 problem. Now that I seem to be an expert, I keep getting asked my opinion. I've had to learn a lot about it, and I've learned a huge amount over the past few months. I guess we all have. I guess I'm going to talk mainly about anti-inflammatories, because that's my own area, but I want to talk more generally to begin with. I think these are the big questions at the moment about COVID-19. First of all, how dangerous is it? Secondly, what about this thing called long COVID, persistent symptoms? Thirdly, can you get reinfected? That's a very important question, I think, at the moment. I'll talk about vaccines and then anti-inflammatories. They're the main things I want to touch on this afternoon. So first of all, how dangerous is it? Well, it's a very pernicious virus, as I guess everybody knows by now. And it is in the coronavirus family, of course, and there are 7 coronaviruses that we know of. There are 4 that cause the common cold, and they're pretty benign. They live in the upper airways, and they don't cause, huge symptomology. These ones, SARS and MERS, they go deep into your lungs, and that's a big problem. A much higher fatality rate. It's 35% with MERS, for instance. It was about 10% with SARS. And this one, SARS-CoV-2, the new kid on the block, that's like SARS or MERS had a baby with one of the cold ones. And this is the baby that was born, because it can live up here in the upper airways or indeed deep in the lungs. And this is the essence of the problem in a way, because when it lives up in the upper airways, you can get asymptomatic spread because the person won't have that many symptoms. They'll be infectious. And then some will go down into the lungs and cause all the problems that we know about. And that makes it especially dangerous because it's very hard to then isolate and track it, unlike SARS and MERS, which were easy to put out because of a symptomatic spread. And I first came across this back in January at a conference in Rotterdam. And one of the SARS experts was there, and they were hoping it would be like SARS or MERS. It could have been at that stage a bit more like SARS or MERS. As the weeks went by, it became clear that it was able to spread through with no symptoms, and that became a very pernicious aspect of this virus, I guess. Now, how dangerous is it? Well, it is a pretty dangerous virus. There's no getting away from it. And this notion of, is it as bad as flu, has come up a lot, especially in recent weeks. This is a good bit of data to show you now how much more dangerous than regular flu it is. And of course, it's not surprising in a way. It's a coronavirus, it's not a flu virus. And this is infection fatality rate. And as we all know, it's a disease of older age, really. As you get older, you get much more problems with this virus. And here we see lots of different studies. And this is the IFR over all those studies. And here's flu, so you can see immediately regular seasonal flu. If you're over 40 or 50, it's 13-fold more fatal than flu. If you're younger, it's the same. They all overlap the lines down here. So as you get older, this is a much more dangerous disease in terms of overall fatality rate compared to seasonal flu. So let's put that particular misapprehension to bed, I suppose, based on these sorts of studies. And then the big one is, can you get reinfected? And we still don't know. Here are the other coronaviruses. So with SARS and MERS, they've shown in studies on those two, you can get about 2 years protection actually from those ones, especially SARS, maybe slightly longer for MERS. This is mainly based on T-cell responses persisting. way beyond when you're infected. Now, the trouble is, the other 4 coronaviruses, it's 6 months protection at most. And there's several studies confirming this now. Even recent ones have confirmed that. And what this means is then you will have a common cold. You will get reinfected. Many will. It'll be milder. That's the good news. It's a less lethal disease upon reinfection, which is a good aspect of this. And there's SARS-CoV-2, unknown. See that we still don't know. And there's influenza, by the way, about a 6-month protection there. Of course, measles, there's lifelong protection. So it depends on the virus. Just because one virus gives you lifelong protection doesn't mean that others will. This is partly to do with how it manipulates your immune system. And coronaviruses have all kinds of tricks up their sleeve for manipulating host defense in various ways and then allowing for reinfection later, I guess, is the overall idea. And there are cases of reinfection now. There's over 20. This is a recent review that was done by Kiko Iwasaki, a very eminent Yale immunologist, and she wrote a great piece in the Lancet Infectious Disease, just came out October 12th. Here are 4 cases where reinfection happened for definite. And look at the range. The first person, they were all mild to begin with in the first infection. Reinfection, asymptomatic, hospitalized, mild, and this one got worse. So what we think is going to happen here now is that, be like any biological trait, there'll be a normal distribution. Some will get reinfected for definite and have a difficult disease. Some will be protected fully. Most are going to be in the middle, and you will see reinfection with mild disease. This is very important to get rid of the herd immunity concept. That a natural herd immunity approach will not work with this virus. No immunologist would say go for natural herd immunity because of the risk of reinfection, and that means it persists in the population and becomes endemic. The only way to get rid of this is going to be a vaccine, as we all know, not through herd immunity. So I wish people would also stop saying that, because that just won't work. It's also impossible to protect the vulnerable effectively anyway. So this is the fact of the matter. Reinfection is a reality with this virus. And then, of course, the other thing is the persistent symptoms anyway, and this is called the pain that lingers. And this is a good review in Science recently. There are many symptoms that persist well out beyond the infectious time frame here for months. And these are getting more and more apparent. Things like lack of concentration, it's called brain fog, breathing difficulties, heart difficulties. These are all evidence. It's not clear yet what % of people have these. Certainly younger people can get it. So therefore, again, even though it's benign in most young people, a certain % will become a chronic disease. A lot of work going into this at the moment in terms of the basis for this. Is this some kind of autoimmune reaction? People think possibly. Of course, research on this and in general on COVID will give us information for other diseases. And there's many infectious diseases where there are persistent symptoms. Lyme disease is one example. We may learn from COVID how to treat other diseases where there are post-viral or post-infection scenarios. It could be used, the research in COVID then could be useful for that. And now onto the various approaches. Of course, massive effort here. The amount of effort going into trying to devise treatments is huge, given the seriousness of this virus. Mainly, you know, obviously the health consequences, but the economic consequences. We've got to beat this virus. It's really important. And of course, there's antivirals, act directly on the virus. Anti-inflammatories, my area. If you end up in hospital, can you be treated? And can you have a therapeutic effect there? And then antibody-based therapies, they're very active. There's at least over 40 of those now in clinical trials. These are an obvious win, actually, in my opinion, because we're well used to using antibody as therapies for lots of diseases. And we know antibodies to the spike protein will mask it and prevent infection. There's lots of preclinical work on that. Clinical trials are running, beginning to release data. Regeneron and Lilly are 2 companies, for example, with very interesting monoclonals that can be used to treat disease, or could be used prophylactically, and that's the way to shield the vulnerable. Give them antibodies in nursing homes, for example. So a lot of work's going into using antibodies as a therapy or as a prophylactic to protect people. And then the vaccine, very briefly, as you would all know by now, I guess, there's over 150 vaccines in development, amazingly. 11 have come to the front, are in phase 3. Various types of vaccines, as you would all be aware, with other infectious agents, you have different types of vaccines. With COVID-19, they're trying every possible strategy. So a whole inactivated virus, that's Sinopharm and Sinovac. Subunit vaccines based on just single components of the virus. S protein is the main antigen. The Novavax and Sanofi vaccines are in that category. Nucleic acid-based vaccines, RNA, is being used by Moderna and Pfizer-BioNTech. And then we have recombinant viral vectors delivering, you know, parts of the virus, often the spike protein. That's the CanSino, AstraZeneca, Oxford, and J&J vaccines. So it's good because all these different strategies, you know, a lot of novelty here, actually, new technologies, new viral vectors are being used. And that gives you a bit of hope, because the previous attempts to vaccinate against the common cold failed, but that was old-fashioned technology. So the use of new technologies we see is really important there. And hopefully some of these are going to work. And amazingly, Barclays Investment Bank about 3 days ago issued a timeline for vaccines. Now, this could be a wing and a prayer territory. It could be pure optimism, but they did dig into the data a lot. They looked at all the phase 2 data, for example. They've got some access to phase 3 data already and safety data. And this is their projection. By the end of the year, 3 vaccines will be approved, they've said. That's the Moderna, Pfizer, and AstraZeneca vaccines. Middle of next year, 5 vaccines may reach the approval stage. Now, isn't that amazing? So maybe by the middle of next year, there could be 8 vaccines approved. Now, there are unknowns here. Vaccines can fall over for all kinds of reasons. Sometimes vaccines make diseases worse, a thing called ADE, antibody-dependent enhancement. So we still don't fully know if this slide is true, but you can give a percent probability of about 75% on this, they've sort of suggested in other reports. So this is where we're headed. Full steam ahead with vaccines, and let's hope they get there, and let's hope the vaccine is safe, of course, and efficacious. And then finally, before I do the anti-inflammatory bit, BCG is an option. I did a review recently on this, BCG and COVID. We'd worked on BCG over the years. This is interesting. It had been known for decades, actually. This is the TB vaccine, to provide protection against other infections. RSV, influenza, HSV, there was evidence that the BCG vaccine could give you protection against those. And now we wonder, would SARS-CoV-2 be also in this list? Now what's happening here, by the way, is this epigenetic reprogramming of monocytes. It's innate protection. You're getting monocytes primed. And then when you get infected with any of these agents, there's a priming, and then you get a better response. And in many ways, we see this as a bridge to the real vaccine. There's a chance the BCG, there's a phase 3 trial, big one running at the moment. That may work, and then eventually the real vaccine comes along later. That might take longer to develop. So some people are seeing this as kind of another option, I suppose, and let's see if that BCG trial works out or not. There's optimism around it, but we've got to wait and see if the trial actually delivers. Now, for the last bit then, my own work, and of course, COVID is an inflammatory disease. Macrophages, lymphocytes, cytokines, the words that everybody in the street seems to know now, the word cytokine. So we know there's a massive cytokine role to play here. It isn't that different to things like ARDS that you'd be familiar with, or septic shock and sepsis. I mean, there's overlapping symptomology here. There are bits that are slightly different with this one, people think. The coagulation part, and the next speaker's on my coagulation, that might be slightly worse with COVID, possibly. But anyway, it's a massive inflammatory process that's getting triggered here that causes all the problems in the vulnerable groups. Remember, most people mount an inflammatory process or an immune response and clear the virus. Young people, for instance, have a very good type 1 interferon response. They think that's why they're able to fight the virus so effectively. But when this virus gets a foothold and begins to get out of control, then we get all these inflammatory consequences, and then we get all the various symptomology that we see with COVID-19. And the good news is the death rate's going down. Here's a good graph of this, actually. This is from a New England Journal paper, comparing death rates across different ages, April to June. And look at this, the death rate. This is actually survival, beg your pardon, on this graph. So there's increased survival in June compared to April. See at every age group. Now that's because some of the therapies are working a bit. The clinical care has changed slightly, and the anticoagulants must be part of this as well. So we are beginning to win the battle in a sense, because it's not as fatal a disease as it was. The second reason of the age is important. It's not just because of young people, because older people are living longer as well. And or at least the fatality rate's gone down in the older people too. Another reason why this is happening, we think, is the dose of the virus might be lower now in the community because people are wearing masks or adopting distancing measures. And when you get infected, you're picking up less of a dose. And that can be very important because it's very dose dependent how bad this disease gets. So low dose is good because your immune system get a handle on it. You're less likely to become severe if the dose that you're taking in is lower than it would have been, say, back in March or April when we're all, getting high dose infections happening. So it's good. This is all good. This is good news. The fact that this death rate is going down. And dexamethasone is one of the therapies that definitely works. Here's a New England Journal. This is people on ventilators. There's the usual care fatality. Here's Dex. And it probably decreases overall mortality by about 20–30%, which is a good thing. Now, that's only in the severe end. It's the ones that have reached the severe part of this disease, but still that gives us hope. And what's happening now is many anti-inflammatories are being tested in the same way as dexamethasone. Will we see effects? I was quite surprised by dexamethasone. It had been tried for years in ARDS and sepsis, hasn't really worked. And yet here it is working in COVID-19 in the severe end of the spectrum. And here are all the therapeutic options. There's antivirals, and there's remdesivir, and these are HIV antivirals, these protease inhibitors. They're in the frame. Chloroquine was there for a while. the anti-inflammatories, and then the antibodies. Now, sadly, the remdesivir trials did show an effect, but the latest WHO-sponsored trial, there was no effect with remdesivir. So we're not sure now. There's a bit. This just literally this last night, whether these antivirals will have a role. They probably have some role to play, but there won't be a magic bullet. Chloroquine's gone because the trials have shown it doesn't really work. There was reason to believe it might, because it was shown to be antiviral in other studies and so on. But no, we don't think that's an option. And now all these here, we have tocilizumab that blocks IL-6. That showed some efficacy. It wasn't great, it must be said. GM-CSF is a very interesting target, I think. And there's a hint that might work. So there's many different cytokines being targeted here. And then we get the antibody-based therapies, as I mentioned already. Loads of antibodies, single chain, monoclonals, polyclonals, and even maybe convalescent plasma, of course, that's a bit controversial as well. So massive effort. And day by day, this is changing. That remdesivir, lopinavir trial just got published yesterday, and there was no effect in that trial. The previous trial said there might be an effect with remdesivir. So it's a moving target. We don't really know. And it's a work in progress. I wouldn't say they give up on remdesivir. Gilead have said they saw that latest trial and they're trying to evaluate it now. It could be subgroups of patients might respond. There could be various reasons why one trial works and the other doesn't. But now my own work, I'm going to give you very briefly what we're doing. We work on the inflammatory process at the most molecular level, I guess. And about 3 years ago, we came across a metabolite, a natural metabolite, made in macrophages called itaconate. And it comes off Krebs cycle. And we think it's a really interesting, potent anti-inflammatory metabolite. I'm going to give you some evidence for this now. And there it is. It comes off Krebs. You may remember Krebs cycle from the good old days. A conatate, and the enzyme IgG1 makes itaconate. And it had been shown to be antibacterial. This is back in the 90s. It can stop metabolic pathways in various, like TB, for instance. So there was evidence that it was antibacterial. And then very recently, an exciting paper came out just about 3 months ago now. Some bacteria feed off it. And Pseudomonas, in particular, in the context of cystic fibrosis, actually, will drive itaconate production in the macrophage. And then it's a fuel source. Look at this. It's eating it and making biofilm. So in some situations, itaconate can boost bacterial growth. In others, it might be inhibitory. And we showed in our paper in 2018, it's very anti-inflammatory. And here's one example. There's LPS-induced lethality in mice. Give them itaconate and the mice live so for longer. So that's in vivo evidence that it's protecting against LPS lethality. And then there was a literature on viruses. Now, we had shown it could block type 1 interferon production, which was interesting. Now, that may be a good or a bad thing. There was a paper on Zika virus, another one on flu. This is all very much in vitro based. And VSV, there was evidence of itaconate having effects on these viruses. And here's a Zika paper, just to give you an example. This is a mouse model of Zika virus infection, and it's lethal in these mice. You can see the dotted line there, if I get the pointer. There's the death from Zika. Give them itaconate, and you can protect them, just like in LPS lethality. And of course, we'd seen that paper, and that got my imagination when it came to COVID. And in that paper, the little diagram, there's itaconate being sprayed on Zika virus. Looks a bit like COVID-2, doesn't it? All this gave us inspiration to work on COVID-19. Now, what does it do? What's the mechanism? Very complicated. Haven't got time to go into it. It's made by the mitochondria and does various anti-inflammatory things. It blocks NLRP3, the inflammasome. It limits glycolysis. It'll block succinate dehydrogenase here in the mitochondria, and that will block reactive oxygen production. We showed that as well. So, sorry, I got Max Artem, I'll show that, but we can confirm all these effects. We showed NLRP3 most recently. So it seems to have a global anti-inflammatory effect. What about COVID-19? Well, again, very nicely, there was a literature building up in the last couple of months. When a cell is infected with COVID-2, you get metabolic reprogramming, succinate accumulates, driving ROS, driving HIF, driving glycolysis and cytokines. And we'd shown that with LPS. And what was nice was this paper cited us because we'd shown this metabolic remodeling event happening with LPS in gram-negative sepsis. Now remember, itaconate can limit succinate. And that was interesting because maybe there was a way to block that metabolic change. And then very excitingly, we saw this paper about maybe 2 months ago now, a big metabolomic screen of COVID-19 patients with severe disease, massive metabolomic analysis. And look what they found, and this is from the discussion, and I didn't even prompt this sentence. Interestingly, itaconic acid, a macrophage metabolite derived from cystic chondroitin, was reduced with severity. Now that got my attention, because remember, it's anti-inflammatory. So if it goes down with severity, maybe some of the severity is due to a loss of itaconate. And maybe then you could supplement it and maybe use it as a therapy. And all that was in my mind as we begin our experiments. And because there was antiviral effects, the first thing we did was test it against SARS-CoV-2 replication in epithelial cells Now, this is a model of epithelial cell infection. The cells die in response to SARS-CoV-2 because it's replicating. That 20% survival there with blue line. Yet we gave them increasing concentrations of itaconate, this derivative called 4OY. And look at this, it can prevent cell death in the lung epithelial cells. It's doing this because it's blocking replication. We measured 2 of the SARS-CoV-2 genes, RDP, RDRP, and E. And look at this, these were repressed in that system, both in supernatants because the virus comes out, and in lysates. So we think it's having an antiviral effect and stopping viral replication. That's the first mechanism known. But just like with Zika and influenza, there was an antiviral effect there. So we're very interested in following up on this. And then lastly, then we've looked at ACE2. There's a nice picture. Someone sent me this recently. There's ACE2 binding the spike protein. There's a spike protein going into ACE2 and the cell to be infected. And there was a paper showing ACE2 expression was interferon dependent. So here we see, say you're sensing a virus here with TLR3 that might sense the viral RNA. You drive interferon production, and they feed back then, and through JAK-STATs induce ACE2 expression. Now we then wondered, would our inhibitor infect this? Because we'd shown that itaconate could block type 1 interferon. Remember I said that earlier? And then we wondered would itaconate block ACE2 expression? And first of all, we could confirm ACE2 induction is interferon dependent. These are in epithelial cells again. There's poly-IC driving ACE2. You can block it with a blocking antibody to the interferon receptor. ACE2 expression, there's ISG20, a standard interferon gene. So we were able to confirm that indeed ACE2 is partly dependent on type 1 interferons. And then of course we tried our itaconate, and in Newly cells, they're a standard human cell line used for COVID-19 studies. There's poly-IC driving interferon beta in those cells. And look at this, 4OI could block that very nicely. And there's a TLR7 agonist, another RNA sensor, again blocked by 4OI. And then of course the killer experiment, lo and behold, 4OI can block ACE2 expression. And here's interferon alpha driving ACE2, blocked by 4OI. There's interferon gamma, again blocked. Here's a Western blot to show inhibition here. You can see the blot there showing inhibition of interferon alpha, for instance, induced ACE2 expressions. That was very nice because we could show ACE2 then could be blocked by itaconate. And then here's a very nice piece of everything. There's poly-IC driving ACE2, blocked completely by 4OI. And there we see it again. That's an ISG and it's interferon gene, again blocked. And here's the Western, very nice inhibition. So we do think we can block ACE2 expression with itaconate. Here is what we think is going on in this particular case. Again, there's a TLR sensing the virus, driving type 1 interferons. They feed back and promote ACE2 expression. And there we have 4OI able to block all of these events. So this is a work in progress, obviously. But there's a chance here this natural endogenous metabolite is there to put a brake on inflammation during COVID-19. If it goes down, the inflammation will get worse in patients based on that metabolomic study. Secondly, we can now use it as a possible therapy. We've got other derivatives that we're developing. A company welding called Citrix that might be used in humans, and it might be antiviral through some mechanism. And secondly, it might limit ACE2, which would stop the virus infecting cells. So we're in the middle of this at the minute, and we think itaconate may be another option as a possible anti-inflammatory approach to COVID-19 and beyond into other infections as well. So let me wrap up now with a few sort of general statements, back to the general idea again. What's going to happen next? Well, immunity might build up. I mean, there will be some protection, even though reinfection happens. There'll be a low level of immunity maybe building up. You can't depend on it to get rid of the virus entirely, but it will be there and that'll help. And of course, a vaccine, that's what it will do. It'll build up immunity in the population. Even if it's only partially effective, it'll make a big difference. I think people will begin to see hope if we see the first data from those first 3 vaccines towards November, December time. And then therapies. Obviously, as I said, it's getting better in hospitals. Great hope for antibodies. And then our novel approaches, it's galvanizing us to look at more therapeutic approaches. With itaconate as one example. Prophylaxis may be possible using antibodies, or maybe using antivirals if the remdesivir has any role to play at all. So again, you'll shield the vulnerable in that way. And then most importantly, the fear begins to dissipate. I think it's very important that if we get to say December, January, and there is a vaccine showing efficacy, that will lift everybody. Secondly, governments can take a few more risks. They can allow 100 people to meet instead of 6 or whatever it is. So you can see slight loosening will happen because there's the prospect of a vaccine on the horizon, and the therapeutics are getting better in hospitals as well. And it's a less lethal disease. And that's the way I see the next sort of period as it emerges. From a research point of view, of course, we're very interested in itaconate and trying to get more on that. And who knows, that could be an adjunct down the line, not just with COVID, with other inflammatory situations. So we're very hopeful for the future. As you probably know, I'm a glass half full kind of fella. Now, there's Alex Hoofman. He did lots of the work on the ACE2, and Hauke Weiss, just to give them credit, my lab, they're doing all the work on Atacanate and COVID-19. And then very important collaborators, Martine van Hemert in Leiden, Lawrence Listenborg in Leuven, they're helping us with some of the COVID-2 experiments, just to give them thanks as well. We don't work on the virus in my own lab, we work on ACE2 expressions. They do the in vivo and the cell infection-based stuff for us at the moment, although we will be setting up opportunities soon. So thank you very much.